Test one, two, three. Test one, two, three. Okay, this is working. You should be able to hear me. I am John Beckmann, Professor John Beckmann, Dr. John Beckmann, the father of sin, the transgenic mother of sin, your hostess with the mostest. I have to present my promotion and tenure lecture. So this is going to be a practice session for my promotion and tenure lecture. All right, the title of my presentation is Bug Biotech and Other Stuff I Do. I do a lot of stuff in the department. And so it's very hard to think through exactly everything I want to talk about because I've done a lot of work in the last 5 years. The way that I've structured the talk is to try to pull out some of the key highlights and the key findings that have been discovered by our lab. So obviously I am John Beckmann. I'm an assistant professor going up for associate professor at Auburn University in the College of Agriculture. Okay, what do? I teach a lot in the department. I teach a 1-2-1 on even years and a 1-1-1 on odd years. Essentially, that means I teach at least one class every semester, fall, spring, and summer. The classes that I teach are medical veterinary entomology, so like mosquitoes, ticks, and the diseases they spread. Applied biotechnology, which is like an introduction of how to genetically modify organisms. Scientific illustration, which is illustrating concepts for science that go into figures for papers, publications, grants, extension publications, things like that. And I also teach microbiology in biotechnology. So I teach a lot of courses. And a big part of my program is actually this YouTube channel that I've created. So as part of my outreach in my program, I record every lecture that I ever give, and I have select class playlists for all my classes. So for medical veterinary entomology, I have a playlist on YouTube of 49 videos. For biotechnology, I have a playlist of 105 videos. For scientific illustration, I have a playlist of 129 videos. So I'm a big archivist and I believe in databasing everything you do to make you a better person. And so I database and make public all my lectures for anybody who wants to watch them online. Again, I actually think this is an important thing to talk about because it's an expanding educational platform. For me personally, I use YouTube to learn a lot. And so it's great for me to contribute back to that learning community. On my channel, I have about 1.4 thousand subscribers. So to give you some context, the entire College of Agriculture in Auburn Agriculture, who have a department who do this, they only have 600 subscribers. I've got double their subscribers. My daughter has about 1,000, and COSAM, the College of Science and Mathematics at Auburn, has 260 subscribers. So I'm doing pretty well in terms of subscribers. And again, my point in this of mentioning this is that this is a big part of my program and it's a big part of my outreach. And it's a big part of how I get my message of what I'm doing, what I'm interested in, what I'm working on out there as a part of an educational component. Just to highlight some of our successes with this YouTube platform. So I have 3 of my top viewed movies right here. I have, for example, a video on Sanger sequence data analysis, 11,000 views, T7 promoter video, 10,000 views, and just a standard lecture for the very beginning of my MedVet Ent class has almost 7,000 views. My point here is that this is a big accomplishment to have a successful YouTube channel, and ours actually just got monetized. It's a very hard thing to do for academic content. For example, if you do a video with your daughters, dancing with your daughters, you get about 10,000 views. If you take a video of your wife making you coffee set to Elvis, you get 5,000 views. And if you're like my daughter and you create a video about fake hot chocolate around Christmas time, you can get 600,000 views. And my point is that it's very difficult to put academic content on YouTube and get this kind of viewership. So I'm doing great. I'm proud of what we've accomplished, and I'm going to keep doing it because it's very useful. Another reason I like to talk about this YouTube channel is that it's actually becoming a part of my research program. So one of the most recent grants that I wrote for the USDA, Administration of an Awards Program, we talked about leveraging this YouTube channel in that grant proposal. And it's a very unique package that only somebody like me can put together. So I may or may not get that grant, but if I do, that's one example where I have leveraged my YouTube channel to put the word out there in a biotechnological context. I'll also just wrap this up with like a why do this? One, it makes me a better teacher. So when I database my lectures, I can go back and I can listen to those lectures at 2x speed and I can refresh my memory. I can critique those lectures and I could do a better job the next time I teach that lecture. So it's a progressive self-improvement. I study the analytics. So YouTube actually has really good analytics. I don't know if I should phrase it as an influence operation, but that's essentially what it is. If you go to these subscribers and you think about who are these 1.4 thousand people who are subscribing to a channel that's heavy on biotechnology and medical veterinary entomology, there's probably going to be a lot of important people in my field that are subscribing to my channel within that subset. And so it's a very important mechanism of outreach to get your name and get your programmatic content out there. And I think that down the line, that's going to help me. So that's one unique thing that I have that other people don't have. Here's an example of some coursework. Here's my most popular class. Actually, my most popular class is scientific illustration. So here are some of the student portfolio of work that come out of that class. This is an example of a repertoire of things that I teach students how to do. So I teach students how to use color, black and white, Photoshop, Illustrator, and video making too, for communication of science principles. It's my most popular class. It gets high registrations every year. Some things to talk about of why this is important. I was featured twice as a speaker at Adobe Corporation conferences. Some of the students that I've trained have gone on to Scientific Illustration Graduate School. So it's an interesting program that's producing really unique students who become successful. And I've actually trained students who then go on to make figures for their papers and who have gotten book covers and journal covers, etc. It's a plug for that class, why that's important. Another big part of my teaching is this international course. So for the last 4 years, I have been teaching in China this microbiology in biotechnology international course. Over the last 4 years, that program has brought in educational funds of greater than $400,000. And my class is a big part of that program. My class is one of the biggest classes. So this recent year, I had 150 students that took this biotechnology class in Wuhan, China. I was actually invited to travel to China and teach this class in person this year for the first time. So people are starting to finally go back to China. I was one of the first cohort to go back and to teach. So here's a picture of me and Torsten, another professor in the College of Agriculture. We're right in front of the Yellow Crane Tower. This is a letter from the vice president congratulating me or thanking me for my help with the program. And one thing I'll say, it was a really great experience to go to Wuhan, China. They're experts in biotechnology, and I learned a lot from going over there. So there's something to be said for international collaboration and going and seeing how people in a different culture do things a little bit differently. I learned a lot from that process. World leaders in biotechnology. So those are some of the highlights from my teaching program. Let's move on to my research. My appointment is 60% research, 40% teaching. Okay, so here's like a mind map of what I do. I do a ton of different things, and my research has really grown and expanded into many different diverse fields. So my PhD is in entomology, and I got my thesis work on Wolbachia and mosquitoes. So there's kind of like this core set of knowledge, which is Wolbachia, bacteria, entomology, and mosquitoes. And from there, I've expanded my program into sort of 4 different trajectories. So one is CI applications towards poultry science in Alabama, mosquito bite blocking textiles, PGPR-based bacterial insecticides, and AI modeling of cytoplasmic incompatibility. So it's kind of like a blob of everything that I work on. And as I go through and talk about my research, you'll see me navigating these different fields. So let me first just explain Wolbachia in case you don't know about Wolbachia. So there's 5 things you just need to quickly understand the system. So Wolbachia is a gram-negative bacteria. These right here are Wolbachia. It's obligate intracellular. So that means it lives inside of a host cell. So these are bacterial cells living inside of an insect cell. It's maternally transmitted. So if the mother has it, in her germline, if the mother has babies, they pass Wolbachia on to the babies in the next germline. It's a reproductive parasite. What that means is it can manipulate reproduction of insects to do very interesting things. And that's the main reason why we study it, is because Wolbachia can be used to manipulate the reproduction of insect pests. Okay. The fifth thing to know about is it's kind of a unique system because nobody has ever genetically modified it. So it's a very difficult system to do classical experiments with. You kind of have to think of creative strategies to do experiments in model systems that you hope trace back to the Wolbachia organism. Okay, so the main reproductive phenotype that I study, that I'm an expert on, that I built my program off of, is cytoplasmic incompatibility, or what we call CI. Okay, this is a classical picture, a Punnett square that describes the CI situation. What happens is if you have a insect that's infected with Wolbachia, that's denoted as pink. So here there's females that have pink Wolbachia and males that have pink Wolbachia. In the males, Wolbachia live inside the testes, and when the sperm of that male insect is produced, the Wolbachia are able to sterilize that sperm. Okay, and if that sperm combines with an egg that does not have Wolbachia, so that's this cross, those embryos die. Okay, so it's like a toxin antidote system where a toxin gets loaded into the sperm. The other side of this is there's a rescue feature where if the females are infected with Wolbachia, okay, they have Wolbachia inside of the eggs that are produced. So the flip side of CI is that if a female is infected with Wolbachia, she puts Wolbachia into her egg. And these Wolbachia can secrete an antidote that fixes the sperm. And so you get this rescue effect, okay? This is CI. And if you ask yourself from the population perspective, which one would you rather be? Would you rather be a female that does not have Wolbachia or a female that does have Wolbachia? Which one would you rather be? Hopefully here the crowd will say, we'd rather be the female Wolbachia. The reason you'd rather be the female Wolbachia is because it doesn't matter who she mates with. If she mates with an infected or an uninfected male, both of her offspring always survive and they always inherit the Wolbachia because it's maternally transmitted. So she's in a good situation. The uninfected female is not in a good situation because if she accidentally mates with a male that's infected, her offspring will die. So it produces sort of like a selective advantage for females that are infected. And what this does is that if you were to measure the concentration or the infection percent, the equilibrium frequency of Wolbachia in a population over time, it causes the Wolbachia infection to rise and to go to a maximal value. Probably near 100%, although there's some other things that reduce it from being completely perfect. So what this is a gene drive system, and it's also a system that's manipulating the reproduction of the insect. So obviously studying ways of how to use this and how this works would be a very important thing to do. That's what my program focuses on. Okay, so there's two applications of CI. There's sterile insect technique and population replacement. So they're both using different features of the system. So population replacement, or the gene drive, is relying on this sort of rescue feature to drive Wolbachia into a population. And sterile insect technique is relying more on the sterility that you can induce in the sperm. So in a sterile insect technique application or population suppression, you're just releasing sterile males, and that drives down the local population over time. Okay. The counter strategy, or the other thing that you can do, is a population replacement. So that's if you're using the gene drive. That's where you're taking a population of insects and you're causing them to be replaced by a different population that has Wolbachia. So these are both in use. Again, this is used in Singapore most recently, and China, to eradicate populations of mosquitoes or suppress them. And this is being used all over the world to replace Aedes aegypti populations with Aedes aegypti that are infected with Wolbachia because they're less likely to transmit flaviviruses. So this is a very important toolkit in the entomologist's biotechnology kit. Understanding how it works is obviously pretty important. All right, so now that you understand a little bit about Wolbachia and CI, I can go into some of our research contributions. So right after I started my assistant professorship, we were coming off kind of like a peak of just identifying the genes that cause CI, which are here, that's right here. So these are the genes that cause CI. There's three different types of kind of like pyrologous operons. So these are the operons that cause CI. So there's two genes. One is a toxin, one is an antidote, and they kind of work in that manner. And they have a few different enzymatic domains. So one's a deubiquitinase, one's a nuclease, and one kind of has both. And so my role in this research is trying to figure out how these enzymatic domains cause the sterility. I'd really like to know the mechanism of the sterility. And in that way, the reason that's useful is because we can then compare that to other biotechnological controls of insects. And we can really compare the mechanisms and decide what's going to be best, what's going to be safest. If you don't understand the mechanism, you're kind of just releasing things and not understanding how they work. That can create problematic situations. So here are my two early publications describing the enzymatic induction of CI. One of these describes the dub domain, and one of these describes the nuclease domain. And this is one of our first big PNAS papers. So I will just show a little bit of data to set up the biochemical context. So SIDB is a dub. SIDB is the enzyme that causes CI. What this means is if you have a protein X, okay, proteins can get modified with post-translational modifications. One of these is called ubiquitin. So it's a little marker that gets put onto proteins and it causes different things to happen. Being a dub, if you're a dub, what you do is you go to that bond and you cut that bond off. So you're removing a post-translational modification from a protein. Okay, so one of the big early first discoveries of these CI operons was that the toxin behaves as a dub and it's able to cleave ubiquitin. So here's a chain. Here's an experiment that shows that. This right here, this banding pattern here across the gel, this is what's called diubiquitin. So it's two ubiquitins that are linked together. And if you have a dub, it'll cut it here and it will convert it to this smaller molecule, which is single ubiquitins. Okay, so you can imagine you have two ubiquitins, cut it in half, it converts them to monomeric ubiquitin. So if you see this band appear, you know the enzyme is a dub. So you mix these chains of ubiquitin with SIDB, that's this lane right here, and you see this band get produced. That means it's cutting the ubiquitin molecules. Okay, so we proved that SIDB is a dub, yes. Here's where the research in my lab really started moving forward as an assistant professor. Is we really became focused on, okay, we know it's a deubiquitinase, but what is this thing? What's this protein X that it's binding to and cutting the protein off? That's what I try to develop strategies to figure out. The other version of these enzymes that cause CI, the other version is this nuclease, okay? So in this case, you have a sequence of DNA, right? And the SINB enzyme cuts the DNA, okay? That's the other type of CI enzyme. And here's the data that show that. This was from our PNAS publication, shout out Hong Li. So if you look at this, the key one you want to look at is this one here, SINB plus circular DNA. This is an agarose gel, so anytime you see these bands here that's showing DNA, if you mix SINB with circular DNA, it cuts it up and destroys it. It's essentially cutting it up into small pieces where it no longer runs on the gel, okay? So this was proof that SINB was a nuclease enzyme. So again, like, why do I show you this? I just show you a little bit to get a sense of the types of experiments that we do. And the primal goal of this is to figure out what is the actual target of these things, okay? We know their biochemical activity, but what's the target, okay? So to answer that question, what is that target X? In order to do that experiment, we set up an experiment where we have SIDB with a his tag, okay? And if you imagine SIDB, you might imagine it as, something like this, with a structure that grabs onto its substrate in order to cleave the ubiquitin off of it, okay? So we set up an experiment where we made SIDB catalytically inactive. So it still has a structural conformation, but it can't cleave ubiquitin. And the reason that we did this is because if you set up a situation like this, you can set up an enzyme where it's grabbing a molecule and it will just hold on to it, okay? And so then we can use pull-down assays to figure out, well, what are all these X proteins that SIDB is binding to and holding on to? Whatever it's binding to and holding on to, that's probably a substrate target for it, okay? So we set up this experiment and we set it up in 3 different ways. So in one experiment, we have SIDB by itself. In one experiment, we have SIDB with the antidote because the antidote rescues. And in one experiment, we have the antidote. So we're trying to get a map of everything it's binding to and how it changes its interactions when it's bound to the antidote. And then we grind up a bunch of fruit flies and we run those proteins over this column. So imagine all the X proteins are from Drosophila. Okay. We run those over the column, and on the column is SID-B bound to its potential substrates. And then we elute the column and we do tandem mass spectrometry, liquid chromatography, tandem mass spectrometry, and that produces these data sets, these lists of all the proteins that you can detect that it's interacting with. Okay. Some interesting things about this data. So here, the SID-B, this is the SID-B by itself, like what is it binding? So the list of whatever its target is probably amongst this 45 proteins. A really interesting thing is that when you combine SID-B with the antidote, it's no longer recognizing the same set of proteins. It's binding a totally different set of proteins. So that's one reason how we think rescue works is rescue works by the antidote binding to SID-B and then changing its preferences for what its substrates that it's binding to are. Okay, so this is the kind of data that we produced from this. This was an early publication in my early assistant professorship with one of my students, Gagan, and collaborator, Mark Hochstrasser. So shout outs there. Then we were able to repeat this experiment. So we did this experiment first with Drosophila. Then the hypothesis is, well, if you have this list of 45 X proteins that CI targets, the question is that list the same as a list from mosquitoes? So the interesting thing about Wolbachia is you can take Wolbachia out of an insect and you can put it in a different insect context and it will still induce CI. So that suggests that whatever that X protein is, whatever the X protein is conserved. And so you should be able to find that protein by taking that Drosophila list of 45 proteins, producing a new list of targets from the mosquito, and then overlapping that list of Drosophila hits and mosquito hits. And the protein that CI is targeting should be in this intersection of those sets. Okay. So that's what we did with Sheun. Okay. And one of the big outcomes that we found with that was some of these really interesting hits that we're really interested in, like karyopherins, which are nuclear importer exporters, and this interesting protein P32, which is important for histone protamine interactions in sperm. Some of these very important hits are repeating in both Drosophila and the mosquito data. So we are able to produce this intersecting subset list. And our hypothesis is that that contains that key protein X, and then we can move forward to functional genetic studies on those targets to see if we can seal the deal, prove it. So shout out Sheun on this work, and shout out Jasmine on this work who helped us get this published. So quick thing here, my only postdoc that I had for 3 months published 2 papers, and he used this paper to leverage a new professorship position, assistant professorship at Ohio State University. So congrats, Sheun. Really good work. Okay, so again, I study a lot about CI, study a lot about the genetics of CI. Another question, so this was our second big PNAS paper. This was work with Brandon Cooper's lab at University of Montana. Shout out Kelly, shout out Daniel, who's one of my students who worked on this. The reason that we started working, what we wanted to start studying was how does CI collapse or pseudogenize? Okay, so there's this really interesting strain of Wolbachia from Drosophila jacoba. So there's a Wolbachia that live in Drosophila jacoba, okay? And the Wolbachia have that CI operon. And so at a conference, Brandon was talking to me and essentially said, well, can you explain how Drosophila jacoba does not have strong CI? It is very weak CI, if any CI, okay? So how can you explain that they have this functional operon, but they're giving very weak CI, if sometimes even no CI? How can you explain that? And so we started this collaborative project, and the first thought was, well, let's look at the operons in that strain, okay? So when you look at the CI operons in that strain, if you compare WMel, which has strong CI, okay, and W Cuba, which has weak or no CI, or what we call maybe hypomorphic CI, if you look at it, it's got a few of these mutations. And some of these mutations are inside of the dub, which we suspect or hypothesize is the cause of CI. Okay, so when we looked at this, my hypothesis was, well, look at the dub. It looks like it's a little bit messed up. Let's check to see if any of these mutations will weaken the strength of CI. Okay, so we first made these mutations in a yeast model. So we have a yeast model where we express the CI protein and the toxin, and it's actually toxic to yeast. So we can use this as a yeast model. Okay, and when you make if you make the H to Y mutation, nothing happens. So if you compare this is wild type, it's still toxic. But if you make that V to L mutation, which is this one, if you make that V to L mutation, the protein becomes completely non-toxic. So there's actually a SNP point mutation that weakens the activity of that enzyme. So we first did those experiments in yeast, and then we followed up those experiments in Drosophila. So here we made the same functional mutation, that V to L mutation. Here's the wild type rate of transgenic CI from the system. If you make that mutation, it weakens the CI. So we actually were the first to discover like a functional, we actually discovered sort of like this natural point mutation that weakens CI. And then we actually measured the ubiquitination status, and we measured the kinetics, and we were able to show that that V to L mutation actually weakens its ability, the enzyme's ability to cleave K63 linked ubiquitin. So we connected this phenotype that the reason that here's the reason why this paper is so cool, is there was a phenotypic observation, okay, that led to functional genomics experiments in yeast and Drosophila, which then led to biochemical experiments that actually sourced the mechanism of how that SNP mutation is weakening the CI phenotype. So that's one reason why this paper's super cool. And there were a lot of people who were kind of questioning whether the dub is important for CI. In my opinion, this paper really like seals the deal on that. You can't really question whether or not the dub is important for CI based on this data anymore. Okay, more on CI. This is the last topic on CI. Another thing that we're starting to study is how the CI genes move around. So bioinformatically, this is a collaboration. Here's a collaboration with Dr. Joe Gillespie at University of Maryland. Shout out Kyle, Shaywood, and Lewis. What we started to notice is that we were detecting some of these CI gene orthologs in a pathogen called Orientia. The reason that this is interesting is because if a pathogen gets hold of the CI genes, that pathogen might be able to gene drive itself into a population of host arthropods, in this case, mites. So it'd be very concerning if a pathogen which is deadly, like Orientia tsutsugamushi, gets a hold of this operon. Can it use it to drive itself and make essentially itself more endemic in populations of mites? Okay. So in order to study this problem, we really wanted to understand how these CI genes were moving. So we mapped out where the CI genes were in these different strains of Orientia, these pink genes, those are the CI operon, okay? And what you notice about the CI operons is they're always flanked by these transposons, which are jumping genes. And transposon jumping genes are known to be able to carry cargo amongst as they jump. And so the innate intuitive hypothesis was that these transposons might be carrying these CI genes. And so we actually mapped out the IS5 inverted repeat, which is a sequence that it recognizes to grab onto and jump. And we noticed that the inverted repeats for this IS5 were flanking the operon. So it looked as if it cut this thing out and brought it into Orientia, okay? And so to sort of like get some evidence for that or to prove it, we cloned out the IS5, we repaired it because it was pseudogenized, and now we're finally starting to get data where we can actually measure the jumping of the transposons, and the final step in this is proving that they can actually carry these CI transposons. So we've already proved that they can carry cargo. The question is, how big is the cargo that they can carry? That's this data right here. So this is Kyle's work where he cloned out this transposon, and if it jumps, it produces colony-forming units that are kanamycin resistant, and you can simply measure the jumping activity based on the number of colony-forming units you get, okay? So this is plus versus minus control. This is induction of the transposon, minus induction of the transposon plus negative control. Here's another negative control. And then you can actually sequence the genome. So we did our first bacterial genomes, sequenced the genomes, and found the insertion sites for these IS5 transposons. So we're very, 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 very close to definitively proving the actual mechanism of how these CI operons jump back and forth between hosts. So that's a really cool project. Shout out, Kyle. And we also measured the CI rates of that Orientia operon, and we proved that it is active, but I don't quite have time to go over that. Okay, so that was all research right here. I do a lot of other research, and so the first sort of like limb that I'm going to talk about is applying CI to agriculture. Okay, so there are, in Alabama, one of the big things is poultry production, right? And in all the poultry houses, there are these beetles called litter beetles, Alphatobius diaperanus, the diaper beetle, as I call it. The diaperanus beetle. We want to be able to get rid of these beetles. They invade the poultry barns. They colonize the insulation and pupate in the insulation, so they destroy the infrastructure, and they spread diseases. So there are certain diseases, nematodes. So this is a heterachus nematode, and those nematodes carry blackhead disease pathogen called histomonas. So it's kind of like a pathogen inside of a nematode pathogen. Those can hang around inside of the beetle in like a paratenic host. Also, things like salmonella can hang around inside the beetle. So you really want to be able to get rid of these beetles as like a cleanliness issue in agriculture in poultry, okay? And so the reason that this is a beautiful application for CI is because the way that poultry farming is done is you have a farm, and in the farm you have these various sort of like barns, okay? And even sometimes in these barns, there's quadranted like sections. So this is a beautiful application of CI, because if we can take these beetles and we micro-inject their eggs with Wakia, okay, we can get an infected line of Wakia that will be sterile when crossed with the wild type populations that are in the barns. And then all we need to do is rear a bunch of males and release them in these barns, pen by pen, progressively until you eradicate all the beetles from one barn, and then you move to the next barn and you keep going barn to barn, farm to farm. So this is our plan to eradicate the diaperanus beetle in Alabama. And this was actually funded by the USDA for $627,000 in a new AFRI that's starting this year. And this should allow me to win the Dean's Grantsmanship Award for my second time this year. This is a collaborative project with Dr. Hauk and Macklin from Poultry Science, and now Ken is at Mississippi State University. He's the chair of the Poultry Science Department there. And also my student, Rich. Shout out, Rich. Good job. Rich produced a lot of the preliminary data that led us to get funding for this great project. So I'm really excited to start that project in the next coming year. Another thing that we kind of noticed when we started studying this poultry situation, there's a lot of bugs in these poultry barns, okay? And one of the questions that I had when we first started studying this was, the litter beetles are the most obvious thing you see, but there's a lot of tiny bugs and things that fly that are hard to see. And so I think the litter beetles might get blamed for a lot of stuff, that there actually might be more important vectors of disease that are smaller that you don't always see. Okay. So what we wanted to do is we wanted to map out every single insect that interacted with the poultry farms in Alabama. So I think we're the first people to produce what we call an insectome. So we went to poultry barns in Alabama and we did trapping for insects. Okay. And we would trap them in different ways. One, we would trap outside the barn. So if here's the barn, we would trap outside the barn, both in the air and on the ground. And in the barn, we would also trap in the air and on the ground, okay? And what we're really most interested in are the specimens that we find that are both outside flyers in the air and inside flyers. The reason that we're interested in these is because if you're thinking about how diseases go barn to barn and farm to farm, it has to be things that get inside and outside and can fly. Okay, so we set up a trapping strategy where we could identify all the bugs that go inside and outside and also fly. Okay, and then what we did is once we had those creatures, we quantified how many of them there were, And we did analysis for whether or not they're carrying Heterachus and Histomonas pathogens and nematodes, okay? And we actually discovered some potential new vectors. So some of the data that came from that, and this is Maria's work, shout out Maria. Some of the data that came from that are that long-legged flies, Condylostylus, and Bradysia, fungus gnats, and Sphaeroceridae, small dung flies. These are new potential vectors that nobody had discovered before. And these might be the most pertinent vectors because they're the ones that are flying in and out, and they're the ones that are positive for the pathogens, and they can fly barn to barn, farm to farm. Okay, so we're really excited to continue work on these new potential vectors for poultry pathogens. Okay, so obviously I also have some expertise on mosquitoes, and I leveraged this for some funding to try to develop some mosquito bite blocking textiles. So this is a major engineering project that we have in my lab. So this was funded by the state of Alabama by an extramural grant for about $900,000. This was my first time winning the Dean's Grantsmanship Award. This has been the hardest thing I have ever worked on in my entire life. The entire project can essentially be summarized as how to deal with this $150,000 machine that keeps breaking, and you need to keep refixing it and engineering it and optimizing it. It's been the most difficult thing I've ever worked on in my entire life, but we did it. We completed it. So we released a preprint. Okay, our preprint out right now is CNC knitting micro resolution and mosquito bite blocking textiles. Okay, and this is of all the work that I've ever done, this is the most traction. This is the most read article I've ever written in my entire life. On the preprint server, it's got over 1,000 downloads. I've never, ever had a paper like that. So we're really excited about that. It should be published soon. It should be published soon, but right now it's in the peer review process. Here's the mosquito proboscis. What you realize in this D is that most clothing does not actually block mosquito bites. So here's a control of an arm, and you're quantifying how many bites you get if you put your arm in a cage. We tested a bunch of different weaves, and we tested a bunch of knits. Weaves pretty much don't block. So of like this shirt right here, this does not block, which is kind of counterintuitive, right? You kind of just assume that all clothing blocks. Most knits do not block, but there is one knit that we found that did block, jegging. So this was sort of like a very important preliminary screen where we figured out most clothing does not block, but there are some things that do. And then the next question was, well, how do you engineer clothing that blocks? Okay, so then we went into the rabbit hole of how do you engineer clothing that blocks? So we bought this gigantic knitting machine, and we started screening all the different knit patterns that we can make. So these are microscopic knit geometries that you can program into a CNC knitting machine, and it knits out these patterns. It's much more complicated than that. It's actually really difficult to knit weird patterns. But we were successfully able to knit these, and then we tested a series of different knits. And what we discovered is one of these knits, called Interlock, has an inherent property where it's able to block mosquito bites in comparison to the other knits. So we really got excited about this Interlock knit. And then we started researching, okay, with the Interlock knit, how can you make it an even better blocker? You can do a few things. You can increase fiber diameter. When you increase fiber diameter, that increases blocking. You can increase spandex content. When you increase spandex content, the blocking goes down. And you can also make the stitches tighter by decreasing the stitch length. If you make the stitch length tighter, it starts to block, okay? And it blocks more than just one mosquito. It blocks Aedes aegypti, and it also blocks Cerophora, which is sort of like the big major mosquito. It's like the monster mosquito that's like super big. And then the final step of this project was trying to figure out ways to make them more comfortable. So these are all analyses of comfort from my student Alexa. And so then the final question is engineering the comfort. And we're now at the point now where we have a very comfortable prototype, and we're looking for industrial partners to mass manufacture this. And so this was great work here. This was, again, hardest project I've ever worked on. Most authors that I've ever published with in this paper. Shout out to Jim, our collaborator at Carnegie Mellon. Jim has been essential for helping us computationally understand the different knit geometries and coding with the machine. Brian, my student, Kyle, Michaela, Isabella, Richard, Alexa, Natalie. Everybody has worked on this project. I'm very grateful that we've got it to this point. Hang with me. Final research project. Okay. So because I have this expertise in bacteria, Wakia, I felt very comfortable transitioning to more research in bacteriology. And so we started working with these libraries that we have here at Auburn. So Dr. Klepper, who was retired now from our department, he spent his whole career collecting these giant databases of plant growth promoting rhizobacteria from different soils in the world. And through talking with people like Mark Lyles and Zach, I really came to see this library of thousands and thousands of microbes as sort of like a treasure trove of genetic diversity, microbial diversity. So anything that you can imagine a microbe might be able to do is probably included in that library. And so we devised this strategy. I've always wanted to try to develop a new insecticide. This is really my best idea for how to develop a new insecticide. What you want to do is you want to set up a screening platform. Okay. And so we set up this screening platform to screen through these libraries. So what we do is we can take a glycerol stock from the library, we can culture it in a tube, and then we can make these apple juice plates. Apple juice plates are for fruit flies. Okay. And we put the bacteria on the apple juice plate, and we let the flies lay eggs. We transfer 10 of those eggs to the apple juice plate. And then we just come back 48 hours later. We count how many are alive or how many are dead. So what we're screening for is we're screening for the bacteria's innate ability to kill insects. Okay. So we've now screened 500. We've now screened 500 strains. So this is quantified how many larvae is it killing. Okay, and this is with 3 replicates. What you're seeing, 3 reps. What you're seeing here, the dots are a mean. Okay, and so what we're interested in are the lines that can kill 4 or more larvae. Okay, obviously we're very interested in these ones that kill all the insect larvae. Okay, so we run 500 strains. That's only a tiny fraction of this library, a tiny, tiny fraction. But from the first 500, we then take the promising ones, all these red hits that are giving us some killing activity, and then we run them again through what we call a tier 2. So in the tier 2, we're increasing the N. We're doing more and more replicates, and we're seeing which of these are real, which of these are not. Many of these red hits are false positives, but with continued sort of screening, you rule out the false positives and you get stuck with what are real actual killers. Okay. And then the real actual killers, we funnel into tests against actual pests. So the real actual killers, we funnel into a series of tests where the first test is obviously against Drosophila suzuki'i. It's a blueberry pest. The reason we like that test is because it's whatever kills Drosophila melanogaster is probably going to kill Drosophila suzuki'i. So one of our best hits is one of the best killers of Drosophila suzuki'i. So we already have a product that we could give to blueberry farmers to kill the Drosophila suzuki'i on their plants. It's not genetically engineered. It's a completely natural microbe. We tested them with plant bugs. So these are cotton pests, ligus plant bugs, okay? And we've got a few strains that are killing ligus bugs, okay? We tested our tier 2s against aphids. So here's a way we're kind of visualizing the aphid data. We've run all these tests. If it's white, it killed it. If it's pink, it did not kill it. So obviously we're very interested in strains like this that are killing the aphids. And then we funnel those into further tests to get further N with the aphids. Okay, and we're also working on funneling these into a fall armyworm pathway where we can find strains that kill fall armyworm. So this is just a figure showing we're working out the controls at the moment for this bioassay. We're trying to find a good strain that's a good positive control for killing fall armyworm. So this work is Jania's grant. This was funded by the United States Golf Association because fall armyworm is a big pest on turfgrass. And this grant to screen them against the aphids, a $25,000 grant, was funded by the Alabama Cotton Commission to help the Alabama farmers. Okay, and I just want to point out that this is a massive project. I could not have done this project without help from David in getting the USGA funding and working with the turfgrass. Work with Kathy, who's helping get me the PGPR strains. Alana's helping us with the aphid culturing, and Dr. Martin, who's a co-PI on the aphid grant, and helping us with the aphid culturing. So I've just had a lot of collaborative help with this project. You might even say I'm a colossus of collegiality working on this project. So also, I'll just to point out all the students who are working on this. Shout out Rich, shout out Jasmine, Jania, Bella, Jay. Final research trajectory is I started getting into evolutionary theory and kind of rethinking some evolutionary theory at a molecular level. And I started applying that to CI research. So we're going to talk about that. That's also a part of another thing that I do. So maybe a lot of people don't realize this, but a big part of myself is I'm very curious and I'm always trying to improve my education. So as I started here, I started a continuing education program in computer science, and every single semester I've been taking one class in computer science. And progressively now I've taken a lot of classes. I'm almost done with the degree. So here's a trajectory of some of the classes that I've taken here at computer science. And I'm finally at the point now where I'm taking AI classes. So I've taken evolutionary computing, research methods in evolutionary computing, and now I'm in machine learning. And I did this the right way, where I slowly, progressively worked up through all the prereqs, and I really understand computer science on some level. So I'm building my repertoire of things that I can do. I think this is important for my research and the biotechnology program in general. And this has actually led us to a publication. Okay. So this was my first AI publication that came out, modeling the emergence of Wakia toxin antidote protein functions with an evolutionary algorithm. So this is a collaborative research project with Dr. T. Dr. T is the relationship director for Los Alamos here at Auburn. And Dr. T is a real expert on artificial intelligence and evolutionary computing in particular. And Joe also helped us a little bit with this project. Okay, so essentially, like, I don't have too much time to go over everything that we're doing here, but what we're trying to do is we're actually trying to encode molecular models of genes, and we're looking at how SNP mutations changes the amino acid structure and how that amino acid structure might change its functionality. And then we're modeling those in simulations where we're simulating the evolution of those proteins over and over and over for thousands of generations in populations that are the size of 5,000 individuals. So large populations over many, many generations. How do those genes change, and how do those changes in those genes affect their functionality, and how can that functionality be traced back to what we actually phenotypically see in the wild? That's what this paper was attempting to do. Okay, and if you might ask, where is this going? So we call this field that we're trying to develop evolutionary algorithms that simulate molecular evolution, ESME. And the way that you can describe what we're trying to do is, protein strings are essentially written words in the alphabet of amino acids. And you can write very, very long sentences, up to 1,000 or more amino acids long. So the search space of randomly putting together protein strings is vast. Most of those are completely useless, would fall apart, they do nothing. So that's what we call by this sea of invalidity. So the search space of actual protein sequences, is just this vast sea of invalidity. Now, within that search space, there is some kind of an archipelago of islands of functional proteins. Okay, so you can imagine one island is possible proteins that actually function. That's a tiny island in the sea of total protein search space. And what nature has given us is a tiny, tiny, tiny subset of possible functional proteins that have evolved and are extant in nature. So what we see in nature, the point here of this graph, or this figure, of this Venn diagram, the point here of this figure is that what we see in nature is only a tiny, tiny, tiny subset of what is functionally possible. And nobody has ever figured out, how large is this archipelago of functional proteins? What is the search space there, and how many functional proteins might be in that designer landscape that can be brought back through an algorithm that could evolve and redesign proteins? So that's what we're trying to work on with this ESME algorithm. We're trying to develop an algorithm that will learn grammatical structures from proteins that exist and be able to evolve them and mutate them into completely new functional possibilities that are grammatically correct. And that's going to open up for us a giant subset of designer proteins that have never existed before. And what we could do with those proteins is infinite. It's a super tough problem, but it's something I'm really interested in developing for my associate professorship in conjunction and collaboration with the computer science department here. Okay, so we talked about teaching, research, outreach. I'm not appointed for an extension. So that's kind of like an overview of my program. Hopefully I explained stuff pretty well. I'm hitting 60 minutes. I did not hit all the jokes I wanted to hit. But this was a good first practice session. So I want to say thanks to my lab. This was early lab picture, later lab picture, and I still probably need a new lab picture. I've had many students work with me in the past. I'm very grateful for all the students who have succeeded in my program. I'm very thankful for my funders, Alabama Cotton Commission, ADECA, USDA, and United States Golf Association. I'm really excited about the research that I've been doing, and I really think that my program is just getting bigger and better every year. So I appreciate the opportunity to talk to you about what I do. Vote for me.
I shall meet death head on. Nicotine, you've got to get out of here. Beware foul demons and beasts, there are many foes here. The time has come to cleanse my heart. This monastery reeks with evil and corruption. Oh, I hate staining my hands with the blood of foul sorcerers. Eternal suffering would be too brief for you, Diablo. Let the gate be your time to die. I shall meet death head on. I shall meet death head on. Nicotine, you've got to get out of here. Beware foul demons and beasts, there are many foes here. The prime evils are no more.